and so on, you could find that these are, you see the scale here is fraction of an arcsecond. So the game is high resolution, high technology, and so on. And this S2 star orbit, okay, from 92 to 2002, and then it has been done again in 2018. Two rounds were done, and that's after that 2000 round when we did that orbit, so that make sure that it repeats itself and so on. And you had now this orbit is 0.05 arcsecond size. Okay, so it is semi-major axis is now 0.119 arcsecond, which is that is the size. So you have the stars resolved, and you could measure the motion. This is, remember that what John Mitchell said, that it is a non-luminous dark object. You could see by other luminous objects. So these are other luminous objects going around the compact object here, and you can then imagine that there is a compact object here, and the citation still says compact object, doesn't say black hole. Okay. So here is a movie which was released early of these motion of these stars, okay, till 2013. And you see now you're coming to the center of the Milky Way, and these are the stars all going around the same common object center. Okay, and the thing is, the S2 is the closest one, which they've been followed for 20 years. And using that adaptive optics, and then you find. So all stars go to the around that object and so on. And this is again the same thing, S2. And this is actually we released by one of these Nobel laureates, Andrea Ghez. Here they actually show you how you can actually rotate the plane. They added more stars on this one. And now you are going from in to out, so you can see even the outer stars, and they're all going around the same object in the center. Okay. And that is our Sagittarius A star black hole. So I think I have a few minutes left. I will just quickly go down to I was asked where is the role of Astrosat in these things. So in Astrosat, what we are observing is those inner accretion disks, how far they actually reach close to the photon sphere and so on. And what is happening around that thing, and that is where Astrosat is playing a big role. So by looking at the disk with the X-rays coming out through this purple region, this truncation happening at the photon sphere and so on, and this disk which is luminous is XUV and UV and so on, and this is too close, so we don't have the resolution to image these things. But what we can do is use the timing and study and the spectra, how it is being reprocessed. Is it the X-rays which are being reprocessed into degraded into lower energy photons, or the high energy electrons which are trapped around here are through inverse complex. They are actually boosting the low energy photons and producing X-rays and so on. So this Astrosat has a huge number of instruments. All were built in India between 2003 and 2015, and the launch was 2015. And this was an X-ray telescope that I built, and which is working beautifully, producing a lot of data. And this is actually useful for this work. And this is actually the satellite before launch in Sriharikota, after just being to be transported to put in the rocket and so on. So, and this fits in this part of the rocket. Okay. So it's pretty big. Okay, it's 1.5 ton satellite and large dimensions, about 2 meter by 2 meter, 2.2 by 2.2 meter, and so on. And all the subsystems are built in ISRO. So, and this is the actual launch of this satellite in a rocket which is called the Polar Satellite Vehicle C30 on September 20th. And this is what we are measuring. This is our soft X-ray telescope is finding this very, very high variability on time scale of hundreds of seconds. okay, 2000 seconds, very fast. And this is a redshift which translates to distance of billions of light years, further than M87, much further than 87. And we can actually set the limits on the black hole in the center using these observations. And this is the another image of another Seyfert galaxy in NUV, and then we study the NUV variation, and then we also study the far UV variations, and so on. And the same object coming from the center. All this emission is coming from the center. We isolate emission, this variability only from the center, and that's where the black hole is. And then we can study the light, how much it is lagging in days and so on, and that gives us a lot of information about the black hole in the center and so on. Okay, so that's where we are in India at the moment, and there's a lot of students and postdocs working on that, and that's how I'm going to end my talk, and I will take questions. Hello? Hello? Any questions? Thank you.
कहाँ के ऑडियो वाले
माउस Thank you very much, Professor PPC, for this deeply insightful lecture on. Unfortunately, as it usually happens with these digital technologies, the YouTube link was not working, and but we are recording lectures, and these lectures will be uploaded on the YouTube for the access to everyone. Since we are running behind schedule, I think we will not have any questions, discussions further. We will invite the next speaker, Dr. Debajyoti Chakravarti. Dr. Debajyoti Chakravarti is a senior scientist and head RNA biology group at the Institute of Genomics and Integrative Biology, Institute of CSIR. His work includes developing CRISPR-based precise genome editing tools for therapeutic applications. More specifically for sickle cell. Recently, during these challenging times of COVID-19 pandemic, he along with Professor Souvik Maity, emerged as a hero of CSIR as his team developed the Feluda assay for SARS-CoV-2 detection. One of the first commercially approved CRISPR-based diagnostic tests to detect coronavirus disease within 1 hour. That is something very outstanding contribution CSIR is very proud of, and they have been bringing all around. So we will have a question here, Dr. Chakravarti, about his discovery or the development of this technology. The sensitivity of Feluda is similar or sometimes better than the quantitative real-time PCR-based tests, which take longer time, but it takes only 1 hour. Dr. Chakravarti did his master's in molecular biology from West Bengal University of Technology, Kolkata, and completed his PhD from Max Planck Institute of Molecular Cell Biology and Genetics, Dresden. Following a postdoctoral stint at a medical faculty in Technical University Dresden, he returned to India to set up his own lab at IGIB. Dr. Chakravarti is the winner of Trust Early Career Fellowship 2016 and Inspired Faculty Award 2015. Now here is a very special introduction of Dr. Chakravarti. See, besides his academic interests, Dr. Chakravarti is an acclaimed musician who plays sitar and has performed widely in India and abroad. He is Sangeet Vicharad and was winner of the HMV Award for Excellence for Most Promising Talent in 2007. I was reminded of the connection of Sir C.V. Raman because he also used to play Veena. So this is something very outstanding about the scientist to have a different dimension. He has given performances and workshops at Berlin Technical University of Dresden, University of Poland, and University of Leipzig, and India Radio. He was also a member of Pangea Tribe, that is a Dresden-based jazz. This introduction for the student even is having a special significance, as I mentioned, that I was reminded of the interest of so these words, I very warmly welcome Dr. Devajan Chakravarti to deliver his talk, and I hope he will be able to have some time so that we are able to complete the bring the session on track, at least in terms of session on track. Thank you very much. Welcome, Dr. Chakravarti.
Thank you, Professor Tripathi, sir. Am I audible? And I'll also be sharing video so you can with my स्पीकर बंद Okay, so is my screen visible? The presentation? Okay, great. I think I can see the hands. Okay. So I just make it full screen. If at any point you cannot see it or if the screen doesn't move ahead, please let me know because I cannot see you once the PowerPoint presentation is on. So first of all, it's a privilege actually to be invited. I am probably the least qualified among the three speakers, and also to speak about Nobel Prizes, which are, which is so far away in the league that it's just a very coincidence and a lot of privilege that I've been invited to be part of this seminar. Thanks for the very kind introduction. I think I'm just a student of sitar, and that's all, more than anything else, as much as of biology. So I'll today probably share with you a little bit about this field of genome editing and why it has become so special over the last decade or so. What are the prospects that it has, and what it can do in terms of changing the way that we look at that we look at the making heritable DNA changes. So yes, can you see my full screen with the slide? Just once more, I'm asking you if you could just put up your hand and let me know. Okay, good. So, okay, so I just Yeah. So I purposefully, kept this name of this talk as rewriting the code of life because, in essence, the 2020 Chemistry Nobel Prize has been awarded to fantastic scientists who showed the world that, in a way, we are definitely we come with our own set of genes, which is what we bring to this world. But for a long period of time, it was assumed that that is what we are going to stay with. Unfortunately, that dogma seems to be rewritten in a way, and so what the code of life here is the gene sequence of an individual, and it seems that it is possible to actually change what is written in that code, and that's what genome editing gives immense power in the hands of scientists and clinicians to do. So, yes, I think you all know about this two brilliant scientists from Jennifer Doudna and Emmanuel Charpentier, who have been awarded the Nobel Prize in Chemistry. It's quite surprising that it went to the Nobel Prize in Chemistry, but there's a lot of background and reason for that. They are principally biochemists who solved the way the CRISPR system actually works. So that's why there was a chemistry Nobel Prize that was awarded to them. But what a lot of you might not know is that the field of CRISPR doesn't actually have two heroes, but it has several heroes. So the heroes of CRISPR are a list of amazing scientists who are actually stalwarts in this field, kind of giants on whose shoulders these two individuals have stood on and have made these landmark discoveries. And this field has not just started today or in 2012 when the first paper from the labs of Charpentier and Doudna came out, but rather in the 80s, 1980s, when the first time the scientists began to look at strange sequences inside bacteria. Okay, so that's why the heroes of CRISPR in the first line, and somebody very finally put this on Twitter, is actually the viruses and the bacteria and the archaea, because that's where this whole field started from. And in 1980s, that was the first time that people began to look at certain strange sequences which were not of bacterial origin, which was present in the DNA of bacteria, and they began to wonder where these sequences have actually come about from. And there was no similarity with any of the bacterial genes, and they kept on looking for other organisms from where this could have come across. And throughout the 90s, they found out that these sequences very closely corresponded to viral sequences or sequences from bacteriophages. Bacteriophages, you might know in the field of biology, these are viruses which attack the bacteria and destroy them because they put in their DNA inside the bacterial DNA, and then the viruses make multiple copies, and because of these copies that are made, they basically propagate a little bit like how the SARS-CoV-2 infection is also working, but in a different way. It's an RNA virus. In this case, these were DNA viruses. Now, what some of the scientists figured out was that there was this bacteria that was attacked by the virus, and the bacteria somehow managed to keep a piece of or pieces of the viral DNA stored within its genome. It was like a librarian. For example, you have a book, and the librarian puts a catalog of some kind, and then this marks the book for future re-entry. It seemed to be having some kind of ability of recognition. And the people who discovered this, Francisco Mujica, as you can see in the first row, he tried to get this amazing story published in some of the big journals. And like in the field of publication, getting into a Science, Nature, and a Cell actually starts a field. Unfortunately, Mujica did not. It got rejected from all these journals. And he managed to publish it in a very low-tier journal. And very interestingly, that actually pushed this entire field by almost 10-15 years because people did not want to take notice of such a landmark discovery. And they had other things to work on. The Human Genome Project was in full swing, and so on. So that also tells a little bit about the publication game and the concept of high impact from publications, because CRISPR would have been there almost 10-20 years back had this concept of judging publications based on the cover of the journal had not existed. But of course, there was a lot of work that happened, and they were in a Danish factory. They were which was making curd. They found that the bacteria was getting resistant to bacteriophages, and these bacteriophage sequences were present, and people began to establish that there could be some kind of an immunity system like we have in the humans. So when we get exposed to a kind of a microorganism, we develop an immunity because there is a pre-recorded, information that is present in our body, and it seems that the bacteria are smart enough to have similar kind of immunity, and that's where the whole concept of CRISPR came about. And in 2012, what Jennifer Doudna and Emmanuel Charpentier showed was that there are two very big components of this whole process. One is a Cas protein, which is called a Cas9, and there is also an RNA component. And if you put them together in a test tube, you can actually make it go specifically to a DNA sequence of interest and cut that DNA sequence at a precise location. And that was fantastic because that actually paved the pathway for using CRISPR-Cas9 for different applications. And there were actually 2 or 3 different papers around the same time that explained the similar concepts also from Virginia Signes. This year, once the Nobel Prize was awarded, there was a lot of talk also about, there were a lot of other people who the scientists considered that, they were also very good candidates. And one of these, big candidates who kind of missed out, as one can say, is located there in the third row. That's Feng Zhang, right in the middle, just next to Doudna. Feng Zhang is from MIT, and also one photograph to the right basically 2 photographs to the right is John Church from USA, who also around the same point of time showed that it is possible to really take this CRISPR-Cas9 system and put it into a eukaryotic cell or let's say an animal cell and make targeted genome editing. Unfortunately, that did not get awarded the Nobel Prize, but these discoveries, which happened right about the turn of 2012 and 2013, is really the backdrop of everything that is being done today in the field of genome editing and the possibility of doing heritable genome editing. So I take this slide from one of Jennifer Doudna's classical slides on how CRISPR is all about repurposing bacterial adaptive immunity. So that's a prokaryotic cell. A prokaryotic cell could be a bacterial cell in this case. And as you can see on the top on the left, that's a virus that enters and brings in its genetic material, the DNA. The bacteria, being very smart, it takes a small piece of that DNA, a very tiny piece, and actually puts that piece within its own genome. It integrates into its own genome in a specific locus called the CRISPR locus. So when that same bacteriophage attacks the next time, which is shown on the right side of the top, there is an RNA that is transcribed from that locus, which is now going to be corresponding to that viral RNA. It's almost like, it's quite amazing how smart the bacterial cell is. And once this RNA is produced, it complexes with the Cas protein and then goes and binds to the viral RNA, which comes as a reinfection and chops it off into pieces. It is so specific that it only does this for viral RNA, and it is also so timely that only when, let's say, bacteriophage A comes in, the same bacteriophage A RNA is produced, and it is direct, and this immunity is directed against it. So amazing adaptive immunity which is present in a lower organism. We really cannot say bacteria to lower organisms, because almost each of the enzymatic complexes that have the most relevance in molecular biology actually come from bacteria, viruses, and anarchy. So that's the principle, that that you can use a complex which is a Cas9, which is shown on the right part of this cell, and an RNA, which is a single nucleotide sequence. Single stranded sequence actually. This can form a complex and can go specifically and target a region on a DNA, and that's the whole concept of genome. How the bacterial CRISPR immunity works. Now keep that thought in mind as we go on to understand what does genome editing mean and where does CRISPR come in terms of genome editing. So when you say genome editing, there are 3 major aspects of the genome editing that one has to understand. is that, if you want to make a change in the DNA of a cell, you first need to localize that DNA. So if you want to localize it in a cancer causing gene, you have to first make sure that you enter inside the cell and specifically locate that particular gene. Then you add a modification, and then a new genomic sequence has to be added, and that's all. So it's a cut paste kind of a mechanism. Easier said than done, but that is how genome editing actually works. As an alternative analogy which I like to give, it's like a word processor file. You have a Word document, and in somewhere in that Word document there is a mistake of some kind. This mistake in a real life would be a mutation in a DNA. So if you have to correct that kind of a mistake, what you need to do is you need to take your cursor to that particular letter. Let's say it's THE, the is written as AHE, and you want to correct the T, A back to T. So you need to take the cursor over there and you delete that A, you hit the backspace button, and then you put in a T, and then you save the file. This is exactly the same thing that is happening inside the cell. So you use the CRISPR complex, which is your cursor, to move to that specific point. You hit a delete button, which happens because the DNA is broken, and you introduce a foreign letter. In this case, you introduce a foreign gene, which will be the corrected version of the gene that you want to enter. And then you save the file, and the save the file happens because DNA is replicated. So all the progeny cells now actually have this heritable change located in them. And how does this work is what we are really concerned about. As in, where do you do the genome editing? Do you do this in the embryo? Do you do it in the cells of the body or germline? As it turns out, each of them have their own advantages, disadvantages, pros and cons. How do you do it? Do you take out the cells from an individual, do the correction on the DNA, give those cells back, or do you try to make the correction right inside the body itself? And most importantly, what are you doing genome editing for? Are you trying to correct a disease? Do you want to prevent a disease? Or are you making a second generation or third generation of individuals who are, let's say, smarter, have a better IQ, and so on? So these are the questions. A lot of them actually ethical, moral, and less of scientific, that the entire field of genome editing has spawned. Just show a small movie here, what exactly is happening. This is the bacteriophage which attacks a bacterial cell, and it brings its DNA inside. So that's the DNA of the virus which enters the cell. And as it enters the cell, which is in this case, it's actually it has being now there is a Cas protein which takes a RNA component, and then it tries to cut this DNA or chop this DNA off into pieces. That's how the bacterial adaptive immunity goes. There you can see it opens up the DNA, and it will now break this DNA. There will be a cut. There the cut happens. So that's the work of the CRISPR complex. So it recognizes a DNA at a very precise location, and this precision is what gives it so much of advantage, that it can specifically locate and cut the DNA. So I will go ahead. So this is a complex of how the CRISPR-Cas system works, which includes a Cas9 protein, which is shown in blue, and there is an RNA component which is shown as a sequence that you see here as in the red. And that around 20 nucleotide sequence, which is called as NNN, this thing of N. This sequence can actually be changed depending upon the gene that you want to target. So if you want to target, let's say, the BRCA1 mutation causing breast cancer, you would involve 20 nucleotides of the BRCA1 gene. If you want to make a correction in P53, you want to include P53 gene, and so on. And that's as simple as that. So you are basically, it's like a postman who is delivering letters, and every time that you change the address in the letter, this complex can be sent to a different gene in the cell. So that's how simple to design this entire process is. And once it locates the gene of interest, what it does is it breaks the DNA. It makes a double-stranded break. And now a cell doesn't want to remain with a DNA break because that's pretty dangerous for the cell. So what it tries to do is that it tries to immediately repair this DNA. And this repair generally happens in two ways. One is a quick and dirty way where the chromosomes are kind of pulled together and try to be repaired. And the other hand, if you try to put a specific DNA template which has a mutation which is the correct version of the mutation that you wanted to correct, then you can actually introduce this small piece of DNA precisely at that location where the DNA break has happened. And that's how homology directed repair of a very precise mutation correction is done. Of course, that's easy said than done, because for all these three things to happen, you need to have the CRISPR complex, the DNA break, and the new DNA, which is a small piece of DNA that you want to enter at that site, to be present at the same point in a small region of billions of letters of DNA at the same point of time. Almost sounds like science fiction, but in principle, that's how genome editing and gene correction, therapeutic gene correction, is being done currently. Just very briefly, what are the things that are being done? The portfolio of CRISPR and genome editing has now extended beyond biology to physics, to biochemistry, to pure chemistry, to applications which are unthinkable: defense, bioengineering, genetic circuits. I mean, you name it, because it allows you to make changes which are heritable, but most importantly, it is a platform, and a lot of innovations come when there is a platform. For example, you can make a second progeny line of primates which have a change in one of their genes, and you introduce that CRISPR complex in the ovum, such that it becomes heritable. So the new primates that are born, in this case these monkeys, are actually having a gene, let's say, resistant for a particular disease. So primate engineering, very, very early stage, it has already been achieved. You can screen the entire human genome, all the billions of letters, for possible drug candidates, for possible response to the SARS-CoV-2 infection, for example, by screening all the 20,000 genes using CRISPR libraries, CRISPR where every gene would be blocked or every gene can be knocked out. Huge scale studies, every RNA can be edited. I mean, RNA editing is, of course, another way by which CRISPR complexes have been used. So again, these are kind of applications that pushes the way we look at personalized therapies, because you can imagine that such kind of drug screens can actually be done at an individual level. If you take your DNA out and then you look at how the azithromycin drug is suitable for your genetic sequences and the mutations that you hold, a CRISPR screen can be carefully designed along that questions. You can illuminate the genome because it ultimately goes and binds to specific regions of the DNA. So if you add a small light molecule or light emanating molecule such as a fluorescent probe to the complex, you can see, let's say here, the telomeres, the ends of the chromosomes, lighted up as these green dots or the red dots. So that tells you that you can specifically bring the CRISPR complex right to the ends of your chromosomes, or for that matter, any gene that you want to. And you can even make genetic circuits where you can basically control how a gene is expressed, and then make that gene as input material for another gene on a different chromosome. So these are like, you have these computer circuits which has the AND gates, OR gates, NOT gates. You can precisely do the same thing inside the eukaryotic cell. Say, for example, you want to make a cell to auto-kill itself if there is a mutation in, let's say, one of the cancer genes. So one can actually try to make sure that that mutation is detected and a kind of a toxic gene is expressed, and that cell kind of goes into auto-killing. It's a huge area for synthetic biology, which is being explored particularly for cancer medicines, where you can, it's because, most of the cancer drugs have got a lot of side effects. So how to build such kind of circuitries inside the cell, so the cell takes a decision: if I am cancerous, I am going to go into auto-killing mode and basically eliminate myself. So such kind of work is also being done using CRISPR. Of course, when you have a technology so powerful, there are a list of things that you can potentially do, and then there are a list of things that you can potentially do, but you should not. And that's why that is the ethics question, which has been highlighted a lot in the last year or so, when Hyacinth Cui was accused of doing the correction in the embryonic genome editing. So at the moment there is a moratorium on embryonic genome editing, where scientists across the world have kind of an unwritten code that they are not going to use it. The reasons are many. One of the reasons is that still the safety and the efficacy of a CRISPR genome editing event is not fully understood. For example, there are a lot of studies which say that CRISPR editing not only puts off-target effects but also breaks chromosomes, and therefore you never know till a long time follow-up what happens to the babies who have these edits. And secondly, it's an ethical question that you are correcting the genes of a newborn, whereas that individual, when he or she grows up, really didn't have a choice. It's in the end it has been done, let's say artificially, but the person doesn't have a free choice to decide whether such kind of editing is necessary or would be required inside the body. I mean, we as scientists often feel that getting rid of this gene might help make you, let's say, resistant to HIV. But the same gene that he corrected would also make these babies exposed to something like the West Nile virus, for example. So these kind of questions are ethical questions, and that's why there is a huge debate. There are international laws in India as well. There are some drafts of regulations as to who owns the rights for actually editing human genome in the embryo and so on. But needless to say that the scope of applications for CRISPR are actually infinite, because it's there in almost every field, whether it's material sciences, in food, in gene regulation, drug development, gene therapy, etc. And that's probably the area where there is a lot of research immediately is how to bring a CRISPR editing into the domain of health, which is correcting a disease, correcting a life-threatening disease. Personally, there are hundreds of mails that come nowadays, particularly after the diagnostic applications and the work that we have been doing, from parents of rare diseases, for example. And in fact, today, just in the afternoon, I'm also presenting at Ashoka University, where there is a big parent group who are advocating the need for similar kind of technologies for correcting, let's say, Duchenne muscular dystrophy, or let's say GME myopathy. Now, these are rare diseases, but at the same time, in an Indian context, a rare disease, because of the population, is actually very prevalent. And some of these diseases are very, very crippling. A DMD boy, it happens only in male individuals, doesn't, gets wheelchair bound by the age of 12 or 13, and has high mortality rate, so lives maybe till the age of 30 or so. And the parents really want some kind of a cure. CRISPR offers the possibility of a permanent cure. I mean, there is no second doubt about it. It is no more at the level of science fiction. But at the same time, we are not at the stage where we completely understand the safety of doing a CRISPR trial in humans. Some countries have started a lot of work in this. We are still a little bit behind, I would say considerably behind, hence the need for more and more discussions. So the question then comes, that there is this blanket laws which say that you're not supposed to do genome editing, and you take your time to get this more and more precise. The question comes that as a parent of a child who has DMD, what decision do you take in terms of trying a new technology? It's not a decision that you would probably want to take at any given point of time. So there's a lot of need for research and development, but also at the same time a lot of dialogue and discussion between policymakers for bringing such innovative technologies. And the next stories that I'm going to talk about, there are two stories that I'm going to tell you today, would actually cover both these aspects. And I'm very happy that, in India, there is a forward-thinking kind of approach in certain areas, particularly in this field, where there seems to be some impetus to actually bring these kind of technologies closer to people. Sickle cell anemia is actually a very debilitating disease. It's one of the first diseases that are being tackled across the world with CRISPR genomic editing approaches. Last year, in August 8, 2019, the doctors actually administered Victoria Gray, who was the first patient to be administered with a sickle cell with a CRISPR complex to alter or to basically correct a sickle cell anemia condition. And there are a lot of companies, Vertex, CRISPR Therapeutics, a lot of them, because of the patent things surrounding CRISPR in the US, they have started this kind of work. And the principle is that you take blood from an individual, you correct the cells, and you return that blood to the individual, and hopefully the individual will get cured. And fortunately, this is a news article from this year. Again, it's just very early. It's just 1 year into the trial, and the person does not have any aspect of the disease left in her, at least phenotypically. She's doing absolutely fine. Of course, it would take almost years to do follow-up, 5-10 years to understand what are the implications of it, let's say, in malignancy, in other complexities, and so on. But it gives so much of hope that, we do continuously hear about the concept of off-target edits, breaks, DNA breaks, and so on. But when you put it all in perspective of the fact that inside our bodies, we are having millions of letters of DNA that are getting mutated almost every second as I speak. I'm not the same individual genetically that when I started the talk. And when you have a foreign DNA, which, foreign complex, which also gives rise to DNA edits, how do we put both these two things into perspective and assess the safety of one versus the other, and then come to a logical conclusion when to give a treatment to a person who really needs it? So this is a very, very nice example. But sickle cell anemia is not just a disease of the West, and as you can see in this graph over here, India is actually even redder as compared when you see the number of patients with sickle cell. Greater than 50% of the sickle cell trained individuals come from India, which means that there is a really a dire need to come out with better strategies, better therapies for sickle cell anemia. And like you see over here, it's a point mutation which causes the disease. And the glutamic acid gets converted into valine, GAG gets converted into GTG in the beta globin chain, and the red blood cells, they become sickle shaped and they form kind of colonies. They block the arterial pathways, and then you have complexities such as stroke developing, people dying, and so on. So in 2017, I started my lab in end of 2016 actually as a faculty at CSIR-IGIB, and the sickle cell mission mode was launched, which was something that was pursuant to the something that was monitored by the Prime Minister's office, and they wanted to see if we could reach a kind of proof of concept application for correcting the sickle cell mutation in patient-derived stem cells. So to cut a long story short, I won't go into details what we did, but we figured out that, if we use the traditional Cas protein, which is a Cas9 from Streptococcus pyogenes, it had some limitations. Although it is being used and its high fidelity versions are being used even for clinical trials in the West, everybody knows that there are some shortcomings in terms of its specificity. And we invested a lot of time in the basic research in trying to find out if there are alternate Cas proteins which might have a high specificity. How we came to Francisella and Novicella is totally serendipitous. There is no credit involved. We have been, we were looking for a Cas which could potentially cleave RNA. There was a paper which said FN-Cas9 could do that. We started working on it, and then we found out all these things. A project assistant in the lab, she was there for a short time. She one day just came and showed me this gel which you see on the left bottom. That if you compare Streptococcus pyogenes Cas9 versus FN-Cas9, on off targets of a target. So OT1, OT2 are off targets, whereas the Streptococcus pyogenes would actually cleave the wild type sequence and the off target sequences. FN-Cas9 will not cleave the off target sequence, and these were just genomic sequences that had just a couple of mismatches, and that was startling because we felt that wow, that's a naturally occurring Cas9 with such high specificity. Why don't we try and see what it can do in terms of genome editing? And that's where we started working. We found that it has a different off-target sensing mechanism where it doesn't bind the target DNA when it encounters mismatches, and we showed that you could correct the sickle cell mutation in Indian patient-derived induced pluripotent stem cells. This paper came out in PNAS last year. It was highlighted as one of the major papers in that issue. But if you ask me, this is more academic than anything else, and one really needs to think how will that help in going ahead and going into clinical trials, and how much of correction do we need to actually correct the disease, which is about 30% to 30% for sickle cell anemia. Since this talk would probably go on YouTube, so I actually removed a few slides which have some IP related things. So I won't discuss more about how we have solved that problem. But we'll go straight into the story of Feluda. So this is something that came about during our work that we started with the FN-Cas9 protein, and work that I have been doing very closely associated with a fantastic nucleic acid biophysicist from IGIB, Dr. Shobhit Maiti. And when you have two Bengalis working on a similar problem, everything becomes very, very Bengali centric in terms of the naming, and that's how the Feluda name actually came about. It's a CRISPR diagnostics, which meaning that you can use CRISPR not just for genome editing, but when you have a Cas protein that is so specific, you can also repurpose it to distinguish between two pieces of DNA which are different by a single mismatch. And that's what we were trying to do for sickle cell anemia. And we know that sickle cell anemia has a point mismatch. So you can actually make the Cas9 protein recognize a wild type gene or a sickle cell gene and accordingly give rise to different types of readout. It could be an agarose gel, it could be a capillary electrophoresis, it could be a fluorescence based readout. And as I will show you later on, even in the form of a paper strip, like a pregnancy strip. Why Feluda? Feluda, the full form is FN-Cas9 editor-linked uniform detection assay. CRISPR diagnostics has started in a couple of years back, with some amazing work done by MIT and Berkeley, again, coming from the labs of Jennifer Doudna and Feng Zhang, where they have used CRISPR proteins such as Cas12 and Cas13 to come up with CRISPR diagnostics which rely on collateral cleavage of reporter molecules. And they call these technologies as Sherlock and Detector, and then there are also similar kind of technologies from China calling it Holmes and so on. So we wanted to give it an Indian flavor. We wanted to have a kind of the name of an Indian detective, and that's how Feluda came about. And probably the Bengalis in the audience know that it's a character from a Satyajit Ray novels and movies. And it works on the principle that if you very precisely put a point mutation in the sequence of the RNA that goes and binds to DNA, then you can specifically distinguish between what is the substrate, whether it's a sickle cell substrate or whether it's a wild type substrate. As you can see over here, if you have this particular combination of mismatches, this is all there in the public domain. So there is, it's precisely at a 2 and 6 position. Then you can make the protein not cleave a wild type substrate, but cleave a sickle cell substrate, and that's what we are using to distinguish between any DNA or any RNA of interest. And really, it is any DNA or any RNA of interest because you can convert this into a technique which can not only work using fluorescence. So what you see on the left is you have a detector which is working on the principle of an affinity base. If the Cas protein is now recombinant, it has a fluorophore associated with it, then you can make this recognize that fluorescence. So you can use a plate reader instead of a gel. And you can also do it for zygosity. Zygosity is, of course, identifying carriers of a mutation, and that's very important because if you can identify a carrier, you can actually genetically counsel that individual to not marry another carrier. I will come to the importance of it in the next slide. We recently figured out that you can do this for more things than just human diseases. For example, with Dr. Govind Makharia at AIIMS, he came and told us one day that patients who come to AIDS at the gastroenterology department, sometimes they come with infection of Helicobacter pylori, which causes stomach ulcers. And several times there are certain mutations in this Helicobacter pylori strains which make them resistant to rifampicin, which is the first line antibiotic. And what doctors do is they take a scrape of that, then they culture it for 3 days, and then if it doesn't grow on a rifampicin, they know, or if it grows on rifampicin, they know it's a resistance variety, and then they give an alternate drug. But not when they come into the OPD. In the case of Feluda, you simply take a strain, get out the DNA, and within an hour, you know if that bacterial strain contains that mutation. So that puts the whole detection procedure almost there at the OPD. This is where we are trying to go towards more and more. And like I said, that it was one of Dr. Shobik's dream projects to not keep a CRISPR technology like this just at the level of the lab. And that's why we wanted to explore what are the possibilities of really doing this on the field. So, this is in a camp in Chhattisgarh, in a village in Raipur district in Chhattisgarh, just before the corona pandemic started. And normally, the government set up these camps to identify carriers of sickle cell trait, and they do a crude test for the solubility test, where you prick the blood and then it coagulates, and then you have an idea that it could be a possible patient or an individual. It's very, very subject bias driven. And then if you have to sequence these individuals, it would now go to the nearest sequencing center, which could be more than 2,000 kilometers away. Maybe it would go to CCMB, which is in Hyderabad, because that's one of the partner institutes, CSIR, and so on. So as you can see, it's not a very easy thing to do. By the time that the results come, and if they want to get a sample again, the individual would be not traceable anymore, because it comes with a lot of stigma to be associated with the monogenic disorder. And if you see on the right, this is a primary school, and the kids are waiting there. And this tiny kid here without shoes, she doesn't seem to be too thrilled at the prospect of having her finger pricked to get some blood out. So what we wanted to do is to come up with an idea where the kids can spit into a small tube, totally non-invasive, and this tube has got certain buffers, all taken from classical Manhattan textbooks and so on, and then it opens up the cell, and then you have the DNA coming out. And then the Feluda reaction can be done within half an hour, 45 minutes, and whether you have that mutation in your DNA or not. So fast, affordable point of care. That's where we are, also trying to do in the next phase of the sickle cell anemia project. Once the students of the lab, they really love this whole experience of going to the villages and actually doing this, like doing a CRISPR test on the field. I think that's what has been the most motivating part of this entire process. Of course, then the pandemic struck and the world turned upside down for most of us, and we thought that here was a detection system that we were doing for monogenic disorders, which had a point mismatch. But if you do it for a viral sequence, it would probably be much more efficient, because here we have 20 bases of match, and that's how the Feluda assay came about. Where you have the RNA from an individual and from the saliva basically, and then you extract the RNA. You do a single step of RT-PCR. Here, RT doesn't stand for real time, but rather for reverse transcription PCR, and then you compare the CRISPR biology with paper strip chemistry to have a readout like this, where in a negative strip you would not have any band, in a positive one you would have the test band, and you will always have a control band which tells you that the experiment actually worked. It has 3 layers of specificity, because it has forward primers, biotin label, reverse primers, biotin label, all against the SARS-CoV-2, and also the guide RNA of the high specificity of FN-Cas9 coming in as well. This entire process can be done within an hour, and it's economical and doesn't require a specialized instrument, except a PCR machine, which is currently something like 20,000 to 1 lakh. There are handheld PCR machines which can come even cheaper than that. This is the first version, which is the version that Tata Medical and Diagnostics is taking out. I will also show you the next versions. We do not even need this PCR machine. And I won't go into details, because I think into the chemistry, but suffice to say that we are using biotin streptavidin chemistry here, and marrying the CRISPR biology with it to come up with this whole idea. which when you put it on a strip, this is how it looks like. So this is a positive control, a positive sample. And if you do not have a SARS-CoV-2, it's highly sensitive. I mean, I think when the DCGI approved it, they got a sensitivity of 96% and specificity of 98%. We regularly, when we do in the lab, we can capture CT values as high as 39 and 40, which is actually beyond the clinically relevant viral loads. And if you're a Bengali, you know that if you have a Feluda, there is also Topshe, who is his cousin, and there is also Jatayu, who was the friend in all the Satyajit Ray movies. So we were on a naming spree at that point of time. We really wanted to come up with a complete Feluda trio. So we teamed up with the Chennai-based startup to develop a smartphone app which would be called Topshe, True Outcome Predicted from Strip Evaluation, which takes a picture of the strip and then it's machine learning trained, so it can give out a readout based on the training that it has, whether it's a positive and negative. It's highly sensitive, like you can see, just a few copies of the viral RNA can be detected. And because we really wanted to make this open source so that other people can design primers for other genes and just use it for your own applications, we had something called a Jatayu, or it's a web server which is junction for the analysis and target design of your Feluda assay, where you put in your sequence of interest, whatever gene that you want to, and you would get a readout as to how to design the Feluda assay. Timing, I have already said, it's cheap. I won't, I won't be able to tell you what is the exact market price. At the lab level, this costs less than 500 rupees. When you compare that with the 2,400 rupees that a qRT-PCR test costs, it's several-fold lower. And I can actually tell you that even that is high because we import the strips from abroad. If these strips are made in India, then this entire cost of a COVID test would be close to 100 or 200 rupees. It can be made to that point. And like I said, that you can do this without instruments now. This is the second version, and the third versions I will show you in a while, where you do not need a PCR machine. We couple this with isothermal amplification, recombinant polymerase amplification, and then just a heating block in a one pot. The entire Feluda reaction can be done. There has been a lot of media attention over this. It's been featured in many places, and now recently ICMR has issued an advisory for this to be a replacement, a possible replacement for the qRT-PCR. That's a big step, I would say, because even the ones in the US which rely on CRISPR tests have got an FDA emergency usage authorization. So it's not an approved test. In the case of India, it is actually an approved test. And it's actually the world's first commercially available CRISPR-based paper strip test. I'm aware of there are other people across the world who are also trying to bring these kind of tests. So just the last few slides, trying to, a little bit of knowledge about what we learned during moving from an idea to a product. I think it was a huge learning experience for me. I'm just 35 years old. So for me, where I had thought that I would probably just focus on the science, and then there was this pressure to not just be there on the bench, but really to try and cover all the hurdles to come into a product format is such a big learning curve for me that, this is probably a good time to share some of the basic facets of it. This is the timeline of Feluda. So idea was the inception came in January, and I can tell you already that that was before the first reported case of coronavirus in our country. This is the evidence for that. As you can see over here, this is January 28th. I made a group. And it says that there is no novel coronavirus cases in India so far, says the Health Ministry. So we decided to form a small group of people who would work on this particular project, and we got into a prototype which was working by March actually. So this was with synthetic RNA, synthetic viral RNA, and we showed that if you dip it into the tubes, you would first have this control band developing. And this was one of the first positive cases that came to Delhi from National Council of Disease, NCDC basically, Center for Disease Control, NCDC, and we were very kind to get samples from there. And we found that yes, we could see this band here. But yeah, this is all works in the lab. You can do thousands of times in the lab, but what are the changes that need to be done to make it work every single time at the hands of a consumer? That's where we spent the next 6-7 months. So we filed a patent on 27 March, licensed the technology to Tata on 6th May, and of course on September 19, DCGI approved it for market release. And this was the the information that came in the public domain. Now, one of the things that I learned is that we speak very different languages. The companies, they speak a different language. The scientists speak a different languages, and you need a middle person to have a dialogue. In our case, the middle person was actually Dr. Anurag, who was the director of the institute. Who literally, how to say, guided both the parties together. Because an IT salesman like this cartoon shows, they always want things to be risk based because that's attractive. It's technology driven because that's how you stay in the market. Very short time to market because you need to be the first person to bring it in time and in budget. And sometimes you feel that that's almost impossible to achieve. And we tested thousands and thousands. These are 2,000, 3,000 samples that we have tested over the last 4-5 months. And we learned a lot of things that science could be very good behind it, but ultimately, if it has to be a product, there are so many issues which needs to be resolved, such as the quality of the reagents that you're getting. For example, a small thing such as a biotin label primer that comes from, let's say, a big company like Merck versus a small company which is a startup could have a huge difference in how you get a readout. The quality of the RNA that the labs were generating from the patients. In certain labs, they were using very, very substandard kits. In certain labs, they were using good kits. Even that affected how the final readout would be. But most importantly, what we realize is that how do you decide a particular new test, in this case a CRISPR test, would be approved or not? One, because we do not have another CRISPR test to benchmark with it. So therefore, there was a lot of discussion as to what would you compare a new test with. And two, the current CRISPR qRT-PCR tests, although they are considered to be gold standards, they are not even really close to gold. Sometimes they are even bronze or even worse, because there are kits in the market, qRT-PCR kits, which perform with 40% sensitivity. So if you now benchmark your new technologies with such kits, then you can imagine what are going to be your sensitivity, specificity percentages. So it's a lot of back and forth, a lot of statistics, a lot of policy making, angry mails exchanged, regulators, non-regulators, everyone involved. But the good point is that, what I also learned during this time is that our students probably are our biggest assets, because these are the people who actually stayed back during the pandemic, decided to be there in the hostel. Almost, I can tell you now that almost every third week or fourth week, there would be a couple who would turn COVID positive. If you now look at our, blood, almost everybody is seropositive, because we were, during the peak period of the pandemic, we were there in NCDC working very closely with the doctors and the clinicians who were getting these samples. And they stayed night and day to actually try to bring this. It was a huge motivation that, you can try to bring a technology that can really go and help people. I think that was what is, and they're still doing it. There are the other things that they're doing at the same point. But most importantly, they say that if you have to raise a child, it requires the whole village to come forward to raise a child. If you want to bring a product to the market in such kind of a time frame, in this case was about 100 days, then you need the entire organization. So not just IGIB, but the whole Council for Scientific and Industrial Research to come together, whether it is the highest, position there, which is the Director General, to the lowest possible in the hierarchy, such as the person who actually opens the doors in IGIB for the vendors to come and give. So it was a mammoth exercise. Everybody was probably involved. And when you have a technology like this, and the Indian media is, all around that, and I had my media stories which can be told at a later point of time. Just to tell you that social media can be very cruel if people take notice of your work, particularly in a pandemic, and then you would have Twitter trolls keeping on attacking that where is the kit, all this doesn't work, etc., and so on. But then the same social media can turn into a complete boom, and it was very refreshing to see that at some point when the DCGI announcement came, Feluda was trending just below Sri Krishna and breast freedom on the Twitter. And when Shashi Tharoor actually congratulates without using any kind of bombastic English words, that tells you that there is a lot of hope even for doing basic science research if it can reach that kind of an audience. And finally, like I told you that, a pandemic is like a kick in the back. It is not, at normal points of time, we definitely take our own, sweet time to go with our work and so on. This is just an example of an innovation where we try to think about Feluda, where we can actually do this test at home. I mean, it's all fine that you take the sample, do it in a lab. But if you want to bring people back to schools, if you want to bring people back to offices and so on, or at the airports, then you would probably need to have even more point of care. This is just a prototype that I'm showing you, where you would have everything inside a reaction tube. This pipetting step is avoidable because all these things will be pre-pipetted into that tube. That the sample is added to it. So there's 3 steps here, and you basically flick it. You do not need to do a centrifugation step. You simply with your fingers flick it, and you tap it on the desk to make sure that stuff comes down. And then what you do is we figured out that you can actually do the PCR reaction, polymerase chain reaction, inside your elbow or inside the palm of your hand. So you can put it inside the inner elbow, and the body temperature generates the polymerase chain reaction. After 15 minutes, you put it back. You add your dipstick buffer, and then the strips go into this as well. And then you can have a readout within the next 2-3 minutes. So the whole process takes about half an hour to complete. So that's where we are trying to go. I mean, in terms of really making it true point of care, really enabling people to return in the times of crisis. It of course requires a lot of reiterations, policy changes. What happens? Are we actually ready to have a test at home, a nucleic acid test at home? How do we monitor the people who are tested positive? Because in a QRT-PCR, you obviously have it linked to a government portal and so on. So all these things have to be worked out. That's where we are slowly trying to go towards. And that's my last slide actually. So there's a lot of people, of course, when you do work in a field like this, a lot of collaborations, a lot of people both from India and abroad who are working. We are very generously funded from different funding resources. Tata is, of course, a big partner now in all of this, Tata Medical and Diagnostics. I'm happy to report that on Monday, they are going to officially launch this for the world, this particular Feluda kit. And we are also waiting with bated breath to see how it actually does in the real life situation. So with that, I'd like to thank the organizers for allowing me to speak today. And if there are no, you can see if there are questions that you want me to take at this point.
उनका आइडियो देना होगा क्या?
I think the microphone is switched off. I cannot hear.
अनम्यूट कर दीजिए. अनम्यूट करिए.
Inspiring story with all of us. You have demonstrated that the knowledge of the level of the Nobel Prize can be translated into the products which can be brought to the common people.
And I think that's all
inspiration for all of us
those who are
working in
different kind of
sciences.
And
I really congratulate you on behalf of the Banaras University.
We extend
our heartiest congratulations to you.
And also good
wishes that you
impact the quality of life
more and more people.
And wish you
success in the next attempts to bring science to the service of the humanity.
Thank you very much.
Thank you very much. We will move on to the next very important talk. From one virus to another.
Dr.
Devajyoti has done outstanding work for taking care of the COVID-19.
And now here
Professor Samita Das, he has virus.
And therefore, we have
requested him to share his experience about the developments in this field and the Nobel Prize in this area in 2020 in the Physiology and Medicine. Professor Samita Das is a professor at the Department of Microbiology and Cell Biology at the Indian Institute of Science, Bangalore.
And
since September 2018, he is the director of the National Institute of Biomedical Genomics at Kolkata. Dr. Das was briefly trained in the area of plant physiology and biochemistry at the Bose Institute as a junior research fellow in the botany department, and he obtained his PhD from Calcutta University in 1992.
working at Indian Institute of Chemical Biology,
where he studied post-pathogen interaction in Leishmania, which causes the tropical disease of Kala-Azar. He did his postdoctoral research at the University of California, Los Angeles, where he virus interaction. Later, he served the same department as an assistant research virologist. During this period, he discovered a small RNA molecule from Saccharomyces cerevisiae that the yeast and showed its selective antiviral activity against polio and hepatitis C virus. This discovery of his led to the foundation of a company which is called Viracin at UCLA for the commercialization of antiviral agents. Dr. Das later on joined Institute of Science Bangalore in October 1998. His group has been studying molecular basis of translational control of viral and cellular messenger RNA. Much of his work has focused on internal initiation of translation of hepatitis C virus and how to exploit it to design novel antivirals. Professor Das has pioneered the molecular virology research in the country. He has been coordinating a DBT-certificated interdisciplinary Center of Excellence for Research on Hepatitis C. Hepatitis C virus, which involves 8 other laboratories across the country. He has successfully demonstrated his ability to transform fundamental research into translational product, which you have seen, an example in Dr. Devajyoti Chakravarti's research. Several antivirals and BLP-based hepatitis C virus vaccine candidates are in the pipeline for commercialization. His innovative and translational research is reflected in as many as 10 patent applications. Many of them are approved and up for preclinical trials. In recognition of his work, he was awarded National Bioscience Award of DBT in 2005, NAFI Reliance Platinum Jubilee Award for Innovation in Application-Oriented Research in Biological Sciences in 2010, and Lenvexi Research Award in the field of Medical Sciences in 2012, and finally his J.C. Bose National Fellowship from DST in 2014. He is an elected member of the American Society of Virology and American Society of Microbiology, and he is also an elected fellow of all the 3 science academies of the country. Welcome, Professor Das. We have been waiting for long. I think you also might have been waiting for long to deliver this talk. We look forward to hearing Professor Das, please.
माइक बंद ये ऑक्स नहीं आ रहा अच्छा उनका अभी अनम्यूट है ठीक
So can you see the slides? So Anil, can you see the slides?
Not yet.
My microphone is not working.
You can hear it? Yes, they both.
Problem?
Now it's better. Can you put it on the full screen mode?
Yeah, can you see the slides? It's fine. Should I start?
Yes.
All right. Right. Okay. I think you can switch off the other microphone. Please switch off the other microphone. Yeah. Thank you. It's a real pleasure. Actually, I was not getting bored. I was just being entertained with all this fascinating work from the two lectures. It's always an honor and a privilege to come back to VHU and then share my thoughts and the work. I think last time I visited in 2017, there was an RNA biology meeting in the VHU, and I met most of you. So thank you, Anil, for this invitation, and of course, thanks to respected Vice Chancellor, my good friend, long-time friend, Rakesh ji. And, I'm always indebted to Professor Lakhotia. And of course, it's always good to see Jagadda on the dais. So, as I told you, we are privileged to give this Nobel lecture series, and particularly on the day of 132 anniversary day of C.V. Raman. It's always an inspiration to all the Indians. And I would like to talk about the hepatitis C virus. First part, I will discuss the contribution of the Nobel laureates or the work that actually led to the discovery of the hepatitis C virus, and then they got the Nobel. And the second part, I will highlight a little bit of research that my laboratory at the Indian Institute of Science for the last 22 years have been contributing in the field of hepatitis C virus. As Anil said, that I'm actually at National Institute of Biomedical Genomics, but my laboratory is continuing at the Indian Institute of Science, and then we are still working on hepatitis C virus. So I thought, just thinking about the students, maybe I will just give you a historical perspective in the beginning about what we know about what hepatitis as such and from when. So when we look back the history, actually going back to 3000 BC, the Sumerians, they used to say that liver is the home of the soul, and the jaundice is the attack on the liver by a devil named Azau. And in 400 BC, Hippocrates actually mentioned that the bile contained in the liver is full of flame and blood and erupts, and after such an eruption, essentially the patient wakes and becomes angry and talks nonsense. Between 17th to 19th century, there was actually reports for the jaundice epidemic in the military men and the civilians. Actually, in the French camp, there was notice of yellowing of the camps or the battlefield jaundice, it used to call. And there was the clinical term was icterus, which is the yellowish discoloration of the skin by the Greek. And this is because of that, due to the hyperbilirubinemia. And the funny thing is, people used to think that it is all because of the bird, Icterus bulbula, which is the Baltimore Oriole. And the myth was that sighting this bird is supposed to cure a person with the jaundice. So all these things while it was going on, in 65, David Boehmer, he was in search of the genetics association with the disease susceptibility, and accidentally ended up discovering an Australian antigen, which is the cause behind the hepatitis B. At that time, it used to call as hepatitis B. Now, hepatitis, all of us know, can be because of alcohol, because of autoimmune disorder. It could be non-alcoholic steatohepatitis. It could be toxins, other infections which can come from the medications and the drugs. But most of it is actually contributed by a range of alphabets. Hepatitis A, B, C, D, E, F is not there, perhaps failed to sustain, and hepatitis G virus. And there are a couple of other non-hepatotropic viruses which also contribute to the viral hepatitis. And if you look at all these hepatitis A, B, C, D, E, and G, they are, all of them except hepatitis B are single-stranded RNA virus. And as you see, hepatitis B is actually on the top with 240 million chronic infections globally, and hepatitis C is also not lagging behind. It's around 150 million chronic infections globally. So it's a major problem. 1989 it was reported, and it was actually discovered to cause the chronic hepatitis, which leads to liver cirrhosis and leads to hepatocellular carcinoma. And in 2007, they actually found that it crossed even hepatitis HIV cases, which was 12,000, and hepatitis C was actually beating them to 15,000. Now, hepatitis C was actually not detected until 2000 in the blood donors. It was not mandated. So it spreaded into the population silently for over a long period of time. Now, in the hepatitis C virus infection, 75 to 85% cases actually establishes chronic hepatitis, but there are 15 to 25 cases are lucky and they can be self-resolving. Now, of that 75 to 85%, actually 20 to 30% leads to cirrhosis, and of that population, 2 to 7% can end up with the hepatocellular carcinoma, which is the end-stage liver disease. How does it come? It can be because of the injecting illicit drug use. It could be the high-risk behavior in the past, could be 10% sexual activity, and there is most of it was actually blood transfusion, hemodialysis. Unknowingly, unfortunately, people have got it into the population, as I told you, because it was not mandatorily detected. And 2% cases were actually the healthcare employment. And soon, this virus was actually found to be spread all over the world. There are several genotypes. There are almost 7 genotypes now as per today. And there are more than 62 subtypes. And because of the variations in the genome, and it's an RNA virus, and with the faulty RNA-dependent RNA polymerase, it keeps on changing and mutating, and that's a big challenge for any RNA viruses, and hepatitis C is not an exception. In India, genotype 3 is prevalent, followed by genotype 1, but in worldwide, genotype 1b is actually more prevalent. Now, I'll just come to the work of these 3 Nobel laureates, the Harvey J. Alter, Michael Houghton, and Charles M. Rice, and how their research led to the discovery of the hepatitis C virus. Now, as I told you, in 1965, hepatitis B was actually found and discovered. Hepatitis A was already in the horizon, but people found there are a lot of people after blood transfusion, even after screening of hepatitis B, they are developing hepatitis. And they used to call it as a non-A, non-B viral hepatitis. So what Harvey Alter did is actually, he has taken the serum from such an infected person and injected that into chimpanzee and showed that the chimpanzee is coming heavily with the infections with the viral hepatitis. That was a Lancet paper in 1978. And so that was the first report of chronic infection and being more frequent compared to hepatitis B. They named it as a non-A, non-B viral hepatitis, or NA-NBH. And they demonstrated that serum from patients with acute or chronic non-A, non-B could transmit the disease to chimpanzee. And one interesting thing they found, that the virus actually contains a lipid. Perhaps it is somehow uses the lipid for its transmission from one cell to the other cell. And the size was ranging between the 30 to 60 nanometer. That was the report in Lancet in 78. Then in 89, Michael Houghton reported that actually they could identify and named it as hepatitis C. What they did is essentially they have taken the serum, ultracentrifuged it, pellet it down, isolate the DNA and RNA, and then made a shotgun cloning into lambda GT11 cDNA library. This cDNA expression library was actually then probed with the antibody collected from the non-A, non-B patient and tried to see whether any of this clone lights up. After several such screening, they ended up with the clone. It was actually named as 511, which showed that it is an extra chromosomal derived from the RNA around 9,600 nucleotide and found only in the non-A, non-B age, that means non-A, non-B viral hepatitis samples and distantly related to flavivirus. Remember, this is 89, sequencing was not so popular. So encoded protein, they found that it binds specifically to the antibodies collected from the patient sera of the non-A, non-B viral. And this is the Western they showed in that paper, that in the science paper in 1989, that it lights up specifically for the serum from the non-A, non-B viral hepatitis, not with the B and the hepatitis A. So this was actually the first report on this hepatitis C virus. Then come the Charlie Rice, and he tried to see, is that possible to see whether this is the hepatitis C alone can cause the hepatitis. So he tried to clone this virus. How to do that? So he went back to the isolations and cloning of the virus from the infected patient, and that RNA that they have isolated from the virus, viral RNA corresponding to this virus, they found there is a 3' non-coding region which is highly conserved. And now they engineered a clone and they actually made an infectious cDNA with that 3' UTR conserved and showed that if you now use this clone and make the RNA in vitro, infect that RNA to the chimpanzee, that chimpanzee comes back with the viral disease. So that was actually a cloned RNA. Remember, Michael Houghton showed that the serum from the infected patient can lead to the infection in chimpanzee. Charlie Rice actually cloned the gene, the hepatitis viral, the whole clone, and in vitro transcribed RNA could actually show infection, not even in the chimpanzee, but also in a cell culture model. So he used different type of cell lines. The one that was successful was human hepatocellular carcinoma, HUH 7.5. And he can actually then show that the virus replication can be monitored in an in vitro cell culture. And this came out in Science in 1997. So you can see how the research progresses. So initially there was an observation by the Alters lab to show that there is a non-A, non-B viral hepatitis exists. And if you isolate that serum, inject into chimpanzee, and then it causes the disease. And if you isolate the viral genetic fragments and then make an expression library, probe it with that antibody from the serum isolated from the patient, you could actually show that this is the virus that causes the disease. Then Charlie cloned it, actually expressed this RNA in vitro, infected chimpanzee, showed that the chimpanzee is coming back with the disease. Not only that, now you isolate the RNA from the chimpanzee and viral RNA, and you can show that viral RNA can actually grow in a cell culture. This was actually the path that the whole hepatitis C research progressed from 1989 till 1997. So now, since now the cell culture was available, sensitive hepatitis C virus tests was also possible, and people tried to make it now mandatory testing from 2000 for the blood donors and the other high-risk people to see whether they are carrier of hepatitis. Because if some patient actually harbors hepatitis C virus, doesn't even know until 17 years when the virus shows up, but by the time the damage is done. So the elimination of the risk of the transfusion-associated hepatitis was actually now under control. And not only that, now there are effective antivirals like the tenofovir or sofosbuvir which are being used, and the disease can now be cured and raise potential for the global eradication of the hepatitis C virus. So that is actually the contribution. So starting from an observation, then identification, and then cloning and establishing formally that this is actually the infectious agent. And that's how, and not only that, now it has come to a stage that we can start thinking about the eradication of this viral hepatitis. In fact, where hepatitis C, all the researchers usually annually meet, we call it hepatitis C virus meeting. And 2 years back, Charlie Rice only actually mentioned in his talk that we hope that 2020 there won't be any hepatitis C virus meeting. Actually, it so happened this time we could not have the meeting because of COVID. But it is true that we are coming close and people have recognized that now hepatitis C virus actually is under control. Now I will just little bit talk about the science behind this, the virus, the biology behind this virus, and how hepatitis C virus actually comfortably lives in the liver. And that's how I said that hepatitis C virus, their life in the liver. It's a RNA virus. Actually, this RNA virus goes inside the liver cells and makes the uncoats it. The viral RNA translates into, it gives rise to viral proteins. The viral proteins binds to the 3' UTR and the 5' UTR of the viral RNA to make it replicate it from positive strand. It's a positive strand RNA virus. There is negative strand made. And negative strands are made actually as a template to make more and more and more positive strand RNA, and that positive strand RNAs are actually packaged, which we call Bredian assembly, and then after assembly, they releases out and then goes out to infect the neighboring other liver cells. Now, within the livers also, they are very specific and infects primarily into the hepatocytes. It is not very common that it infects the stellate or the Kupffer cells. And it is actually, looks like 80% of the hepatocytes, which is, constitutes the liver populations of the cells. But there are endothelial cells, there are stellate cells or Kupffer cells, which is Kupffer cells are considered the macrophages of the liver, but how they are spared. So, I can imagine it's something like somebody tries to enter into a house. They know how to enter and where actually to go. They actually target goes to the kitchen where they can make use of all the vessels and the spices to make palatable food. But the host, either they are unalerted, they don't know, or they starve outside, and the virus can happily stay and make their palatable food in the kitchen. And the food is also very palatable. Many of you like this sub sandwich, and we usually cut it. We can't really handle such a big polyprotein. The virus also does that. This polyprotein is actually cleaved by some cellular and the viral proteases into structural and non-structural protein. There are structural protein like the core and envelope proteins. The P7 is actually ion channel protein. And there are non-structural protein which are actually coordinates different functions of the virus, like NS2, NS3 is the viral protease, NS5A and 5B is the RNA-dependent RNA polymerase, and 5A is the one that actually determines the sensitivity to the interferon. Long time, people used to be treated with interferon alpha, and later on they found, well, pegylated interferon alone cannot really solve the problem. They started giving ribavirin, which is a nucleoside analog, along with it. But the success was only 40% people used to respond to the therapy. Until recently, when we have the pilavir, which is actually inhibits the NS3 protease of the virus, and the sofosbuvir, which is actually targeting the RNA-dependent RNA polymerase of the virus. And both of them can effectively bring down the virus titer. And now there is a ledipasvir, which is NS5A targeting drug, which is also available in the market. But any drug can actually control a disease to some extent, but there will be always issues. The limitations here is a genotype specificity. I told you that hepatitis C has 7 genotypes and 62 subtypes, and it is keep emerging. There is emergence of the drug resistance mutants, they relapse once the drug is stopped. There are a lot of patients after 6 months, although the virus is not detectable, there is hepatocellular carcinoma going on. And additionally, therapy-associated side effects are also there. So, people still are actually continuing to understand the biology of the virus and to understand, is it possible that we can have additive therapy or additional therapeutic approach which will be more effective and will probably bypass the emergencies of the drug resistance. Now, what we have been doing it for last 22 years in actually so happened, this is discovered in 1989, and 1991 I landed up in the UCLA. And I started working on hepatitis C virus since 1991. So it's almost like my research career or experiences is actually going along the trail of hepatitis C virus researches all through these years. But I was actually focusing more onto the hepatitis C virus translation, and I'll tell you why. How we make our protein. As you know, we actually have a capped mRNA, which is 5' capped, and it's actually 7 guanosine modifications, and which attracts a lot of initiation factors binding, like the IF4E, IF4A, and 4G, and that attracts the joining of the 40S along with another sets of initiation factors, and then it actually scans for the initiated AUG, and that's the place where the 60S joins and then starts translation of the open reading frame. This we have read it for a long time. This is the only translation people knew about. Now, from hepatitis C, we found that it is actually uncapped RNA. You don't see modifications at the 5' end. The untranslated region is relatively actually long, which is around 342 nucleotide, and there are multiple AUGs. And ribosome, in fact, doesn't bind to the 5' end. It binds to an internal sequence, which is a scaffold of structure, and that attracts the 40S direct interaction, like a prokaryotic type of interaction. So the 40S can directly bind to the hepatitis C RNA, which we call internal ribosome entry site or IRES, and then that actually leads to the joining of the 60S and then goes for the translation. So this is called cap-independent internal initiation of translation, which is actually unique and fundamentally different. So when I joined this, when I started this lab at the Indian Institute of Science, we were actually contemplating what can be done. Same time, there was a paper in Science, actually Jennifer Doudna was one of them, and with Jerry Frank, they showed that in the ribosome, in the 40S, this is the head, the platform, and the bit. Usually the cellular RNA binds to the A sites and the B sites, but the virus somehow ended up binding to the exit gate of the ribosome. What it does, it actually enters from the back gate, triggers a conformational alteration, and then actually access to the A sites and the B sites. So that the viral RNA does not have to compete with the cellular RNA, which are crowding the front gate, A sites and the B sites, but rather actually makes a contact or bribe the person sitting at the back gate and goes inside the and makes a contact with the ribosome and then gets its purpose done. Since it is unique and fundamentally different, we wanted to understand that which all events are actually important, which all ribosomal proteins are involved in interactions, which all host factors, like other than ribosomal protein, there are several other protein participants in the internal initiation of translation, and which all viral proteins are actually regulating the translation of the virus while the replication is also still going on. So this was actually the theme of the lab when we started 22 years back. And in the back in the mind, we were thinking, is that possible that once we know, can we target them to design selective antivirals? Because this is unique and fundamentally different. And if we target it, we don't expect, although cellular RNA are also known to have some IRES element, but their requirements is quite different. They don't bind to the 40S from the back gate. So what we showed actually, it's my first student who is now a faculty in the ISAC Kolkata, he showed that you actually use different small structured RNA from the 5' UTR of the hepatitis C, and one of them was successful, which actually goes and binds to the ribosomal protein S5. And this ribosomal S5 protein is the one which is sitting at the backgate, and hepatitis C RNA was driving this particular protein. So he actually put them into the same coin. This viral RNA now goes and binds to this, but instead of this, we are using a small RNA derived from the 5' UTR, which goes and sits there, prevents the viral RNA interaction and prevents the translation. This was actually, we got the US patent, and we are trying to commercialize, trying to see whether it can be used as an RNA inhibitor. I was also mentioning such an event also involves a lot of other host factors. Now there are proteins in the nucleus. They have one of such protein is called human LA protein, which was originally found in the lupus erythematosus disease. It's actually LA autoantigen, but in the infected cells, it comes out to the cytoplasm, binds to the viral RNA, melts the structure of the IRES region so that the 40S can come and bind it. So what we did is, since this viral protein was very important, this cellular protein was very important, we tried to study and see, is that possible that we can actually identify the RNA binding motif. We narrowed down to almost 7 peptides, and we solved the structure of this 7 peptide, which is a beta hairpin structure. This was done in collaborations with Siddhartha Rai's lab. He was there at that time, and IICB Kolkata. And we showed that if you use the 7 mar peptide, hook it up with 6 arginine residues, then it can cross the cell membrane barrier, goes inside the liver cells, and kills like a surgical strike the hepatitis C in the liver cells. And recently we have now cyclized this peptide and tried to use this cyclic peptide, which can prevent the human LA protein interactions with the viral RNA and achieve the inhibitions of the viral translation and the replication. So in both cases, actually what we did is we are trying to mimic a situation where, like, the antibiotic prevents the ribosome and the bacterial RNA interaction, and they are successfully doing so, and we are happy with that. And we were actually proposing that you can actually think about preventing the ribosome, 40S ribosome, and the hepatitis C RNA interaction using a small RNA or a peptide, and actually achieve the feat of inhibiting the translation. Another thing I was just trying to say that hepatitis C virus was actually does not infect small animals. Like, it don't have a mice model. It does not infect the rabbit. It does not infect mouse. It does not. So it infects human and chimpanzee. So we were trying to see, is it possible that actually something is missing in mice and rabbit, but it is there in the human and chimpanzee. And we found actually it is true that 7 mar peptide that we made and showed is so important for the LA interactions with the HCV RNA is not conserved in the mouse and rabbit. And we demonstrated that if you now revert it back and make it humanized, then the mouse LA can start binding to the hepatitis C RNA, and it can support translation. So CRISPR-Cas, as Debojyoti explained, was actually not there when we actually did these things. It's not so popular. Now we can think that we can knock out the mouse LA, knock in the humanized LA, and try to see whether we can have a mouse model for hepatitis C virus infection. If we can take care of the transgenic mice which are expressing the other receptors that are necessary for the virus to enter the mice liver cells. So this is something we have been working on hepatitis C virus now a long time, and we showed over the years that several proteins, like I talked about the LA, we have also worked with 2 other proteins. One is PTB, which is polypyrimidine tract binding protein, helps in splicing. HUR, another protein which we are now recently working. We showed that how this protein comes out from the nucleus to the cytoplasm under infection, binds to the 5' UTR, some of them binds both 5' UTR and 3' UTR. And what is interesting is, when they come out to the cytoplasm, they are usually modified, and that's how they come out. They lose visa, they are sent out. Most of the time it is a phosphorylation or acetylation, and these modified proteins have greater affinity or lesser affinity. So we have tried to understand what we are doing now, how their modifications changes their affinity and the cytoplasmic availability so that they can bind to the 5' UTR and 3' UTR and control the virus RNA translation. So it's actually, you can imagine a continuous tug of war between the virus and the host cells. We showed that HUR protein also helps in the virus to win over, but there is cellular MIR125B which prevents HUR to actually do what it's supposed to do. But no matter what you do, there is OLA on the road which can actually tilt in their favor. The whole game is now changed in favor of the virus. But OLA is also under a traffic control, and this is the traffic control we are trying to understand. What determines the abundance of LA and HUR? What determines the modifications of these nuclear protein to come out to the cytoplasm, bind to the viral RNA, and either help the virus or help the cells to win over this tug of war. So overall, to me, it looks like a big orchestra, and with the tune of hepatitis C, they made other proteins to dance. It could be HUR, could be the drummer LA, it could be the PTB flute, or it could be the violin miRNA, or it could be the long non-coding RNA. You know, whenever I just come to PHU for any presentations, I think about Professor Lakhotia. He has been the proponent of the long non-coding RNA for ages. And this is another thing that we showed now, how long non-coding RNA can also change the game. And you know, they bind to the either 5' UTR or 3' UTR, and their relative abundance and affinity decides whether the virus will translate or replicate. Remember, the viral RNA initially translates, the same RNA will be used for replication as well. Ribosome has to make way for the replicase to move on. So there is a lot of circuitries there. There is a concerted effort, and that's over the years we have been trying. Just show a couple of slides on the long non-coding RNA that we recently found by RNA sequencing from the infected patient sera, from the cirrhotic and the hepatocellular carcinoma. We found one long non-coding RNA is the HULK, highly upregulated liver carcinoma, and we showed that how this HULK actually helps to load onto the lipid droplets and increase the lipid droplets, and finally actually helps the virus release out from the VLDL pathway. So we showed that HULK, it's a long non-coding RNA, increases the number of lipid droplets, helps in the loading of the viral protein into the lipid droplets, and not only that, it packages the virus and then sends it out to the neighboring cells. And we also studied the proviral activity. Another thing that we found that hosts also have a antiviral activity for that. They keep the HULK under check. There is microRNA 122 which is sponged HULK and somehow prevents the HULK activity, and that's how they maintain the replication in a low profile, and that leads to the chronicity, establishing of the chronicity, and that ultimately contributes to the development of the hepatocellular carcinoma. But the researches are still going on. The puzzles are not solved. As Debojyoti was also saying, that you show something, then you ended up asking more questions and try to keep solving and make it much more fine-tuned, like they have been doing it with the Felura. So essentially, as I told you, that we have been working on the hepatitis C virus-induced hepatocellular carcinoma, which is still not solved. There are proteins, there are microRNAs, there are long non-coding RNAs, and lately we are now somehow linking it with the P53, which is the dada of the cell, and we try to see how this dada is actually contributing to the hepatocellular carcinoma. Dada has a chota bhai that's called P47, which is a truncated P53, which showed that also have an IRES. That was from our lab only we showed, and we tried to see how they can solve the puzzle. Now, while we were doing it, Government of India insisted us to focus on some of the antiviral and the vaccines, which could be possible to get it from for more terms of the human health management. So we try to use our expertise to isolate the pure compounds from the fruits that we consume almost every day in the breakfast table. And we showed from our lab there are three compounds, punicalline, punicalagine, and elachic acid, which has the activity almost similar to telaprevir. It inhibits the NS3 protease by binding and blocking the catalytic triad in the NS3 protease, which we are now trying to see how actually yesterday I had a discussion with Professor Katia from Imami, trying to see how we can actually make it to the regulatory. As Deputy mentioned several times, that it is a challenge to somehow take this bench side to the bedside, and there are regulatories in between. So we have been working on it. Another thing I just wanted to show you that we have shown a lot of people used to use Bhumi Amla, which is called Phyllanthus amaras. In fact, Tyagarajan from University of Chennai, he has gone to the human trial to show how Bhumi Amla can actually cure hepatitis B and C. But we didn't know which are the compounds that are working on. Very recently, we have actually shown that there are compounds called phyllanthine and corylagine that can inhibit this virus, again the NS3 protease, by inhibiting the catalytic sites. And we, not only that, we have recently showed that how we can make them water soluble by doing some chemical biology along with my organic chemist friend Narayanaswamy Jayaraman at the Indian Institute of Science. And all we are trying to see now whether these are all in vitro culture can be extrapolated to an in vivo system. Now, as I told you, this is a challenge because we don't have any mouse model for hepatitis C, but there is one in Japan where they have actually transplanted human hepatocytes into the seed mouse. Now, these mice are actually dependent on the human hepatocytes and can take up the hepatitis C virus infection. And we have shown that if you use this model and have the infection going on, in that model, if you use our corylagine, actually you can see a level of protection. And this is actually interesting, but it's every shot causes $10,000. So you could not really do much experiment, but this is a pilot thing. What is very, very enthusiastic at this moment is it prevents the collagen deposition, which is central to liver fibrosis. And we are actually pursuing this actively and try to see whether pomegranate, which contains this corylagine as well, can be used as an add-on therapy along with the direct-acting antivirus, which is the telaprevir and sofosbuvir, so that the patients which are reducing the virus titer can also make sure that their liver does not progress into hepatocellular carcinoma. So we worked out the biology behind this activity of the punicalagine and corylagine. We showed which all pathways it are affecting. I'm not getting into the details, but I'll just show one last thing. I want to show that we have been working on the vaccine. As you know, till date, we don't have a vaccine for hepatitis C virus. We have made one virus-like particle called VLP in baculovirus system and in insect cells, SF21, and then we showed actually this baculovirus system can make this VLP, and this VLP can elicit enough immune response, which if you isolate the neutralizing antibody from there, can prevent the virus proliferation. This we have done it in mice, but we don't have any challenge model in mice, not even in pig, but pig immune system is closer to human. So that's how Government of India now given us grants to test it in a large cohort of pig and try to see whether actually it can elicit similar level of immune response so that we can go for preclinical trial leading to the human trial for the hepatitis C vaccines. If it works out, this is customized for hepatitis C virus in India, which is made for genotype 3A. And we are also trying to see if we are successful, this will be the first vaccine because we still don't have the vaccines. So as Debujiti was actually showing how to translate from a simple observation into a translational thing, we started working on translation protein synthesis and showed how many ways you can actually make it translational, be it a small RNA or a peptide or monoclonal antibody. This we actually isolated neutralizing antibody from the patient's sera and showed that neutralizing antibody you can now inject naked monoclonal antibody. Now we have a single cell binding protein set, and then that protein can be used as a therapeutic use. We showed that you can use our technology and then try to somehow come up with the natural sources and isolate the pure compound and try to see whether that can supplement the add-on therapy. And we are also making an effort in the VLP vaccine. All of these things are being tested in the PXP mouse, which is actually humanized liver, and try to see whether we can get something going. So all these things are possible, not single-handedly, but with a large level of collaborations. And this I would try to impress upon the students is, I think it is important that you have a dream. But to achieve the dream, you need to know that you need to take the help of the correct place and correct time. So correct person and correct time is very important. We were lucky to have so many people helping in our antiviral therapy, our host virus interaction understanding, our pathogenesis, which includes Shiv Shireen at ILBS, Rakesh Agarwal, who is now in JIPMAC. We have Shoma Banerjee in IPGME at Kolkata and Yashoda Hospital, Hyderabad. We have a large collaboration going on. And we also work on liver-targeted delivery. All these antivirals that we are making, we are trying to target it into the liver. And we have shown how can be done with Devi Sarkar at Delhi University with the virosomes. We have shown dendrimers through organic chemistry. Recently, we published a nanocapsules, and we showed that how nanocapsules can actually encapsulate your inhibitors and then deliver it directly or specifically to the liver. We are also working on PLP and DNA vaccine. But nonetheless, I would like to say, just to keep your research interest alive, you keep thinking out of box. My out of box thinking is my mechanical engineering friends, and at IISc we have now a program on liver mechanobiology where we try to understand, mechanical engineer is trying to understand the liver stiffness, how the infection of the virus causes the changes in the nuclear shape, and the nuclear shape changes the chromatin and the gene expression, and leads to the laminopathy. And that's how we go. We also work on Ruth Malik in PIFR, and Naren Dixit helps in our mathematical modeling. We're trying to understand whether we can provide some mechanical biomarker, where looking at the mechanical biomarker and biochemical marker, the clinicians can convincingly predict that this patient, although the virus is gone, is likely to progress into hepatocellular carcinoma. So this is something is translational, and I think that is something which we have been pounding on it. And what I'm just saying that hepatitis C virus is now, research is worldwide encouraged because of the Nobel Prize given to hepatitis C virus. It's an appreciation of the effort of the people who have been working on hepatitis C virus for a long time. Definitely those three pioneers, but the story is still not over. We don't have appropriate model for the hepatitis C virus research. Only non-human primate, as I told you, practical and ethical constraint with the chimpanzee use. So now it is stopped worldwide. So not naturally susceptible to hepatitis C infection. So people are now falling back into humanized mice or ectopic liver degeneration. Vaccines, still there is no vaccines, although VLP we are trying, but there are other vaccines, but they are in different stages of trial, but none of them has come to the commercialization stages yet. Now the challenges are because of the huge sequence error rate of the RNA-dependent RNA polymerase in the N-spike, the extensive glycosylation of this envelope protein that also changes its epitope and the antigenicity, and the limited production of the whole inactivated virus. This is another problem. We can't really make tons of hepatitis C virus. Even after 31 years of the discovery, we still can't make it in liters of hepatitis C culture. And that's we are trying to, that is the big impediment into this research progress. We're trying to overcome it. And as I told you, hepatocellular carcinoma, particularly hepatitis C virus-induced carcinoma, what are pathways are involved? how the epigenetic modulations of the RNA or the protein contributes to the hepatocellular carcinoma is still in the card, and we're trying to solve it. There are exosomes which are involved, and that carries virus from the liver to the other organ. In the late stage of hepatocellular carcinoma, it actually invades to the other organs. And we are trying to understand how this exosome and what are the contents that are actually contributing to this. Finally, I would like to end this talk with a message from none other than Charlie. We have been working for a long time in collaborations. So when I congratulated him, he also appreciated that actually the story is not over. Let's hope that this example of the benefit that the science can bring will encourage the public to embrace and support fundamental and curiosity-driven research. This is the key word I would like to impress upon the students. It has to be the question that you are asking. You should be enthusiastically ask that question. That should be a fundamental, simple question, but curiosity-driven. That will keep you motivated lifelong, and at least you can actually, if you follow that, he says that as we are in the midst of experiencing with the ongoing pandemic, there are many challenges ahead, and RNA virus is the one will keep ourselves busy, one after the other, be it chikungunya, be it dengue, be it SARS, or so, we will probably have to understand that we try to ask the real appropriate question and real appropriate approaches to solve these problems. And I would like to thank the group, now the present group. So many people have graduated, they have contributed. It's 22 years of work that we have been contributing to the hepatitis C field. And as Anil pointed out, that we are actually a center of excellence for hepatitis C virus research. There are several laboratories in India are involved, and they are actually contributing to the ongoing research. Thank you.
इनका साउंड जाएगा ना.
So I will now stop presenting.
Thanks so much.
Samudra for a wonderful. You spoke like a master craftsman. Building a making concepts very simple. That's what was the situation of the Vaishnava model. Telling the stories that can collate down to each one of us. I think it was a beautiful talk. And the other thing is very important. The commonality between you and the Chakravarti, that leading such a team to a task, coming out with a determination, vision, and be able to deliver these processes and the task and something outstanding, which requires a lot of and generosity. And I think that this speaks volumes about both of you, leading the and going high and very good science, but translating that science for the service of the humanity. So thank you very much, Samudra, once again for accepting our invitation, and I would like to thank all the speakers for their excellent talks. And there were some technological glitches which I would like to compensate by putting these talks on the YouTube. And I would like to express my heartfelt gratitude to the Director Institute of Agricultural Sciences for giving us this hall, which is big enough to make sure that we maintain very good distance from each other and taking precautions of the corona. And Professor Goenshi is sitting all through, the Director of the Institute of Environment and Sustainable Development, my dean and other heads of the departments and other colleagues who have been here, and those who have joined us online even after the problems. We have been able to generate a second link which has been sent to many of them. They might have joined, and I would nonetheless like to thank my staff from the Institute of Science who have been very helpful and very active in organizing this event. The most important thing which at the end I would like to mention that Professor Jagad Roy is a driving force. He likes to be behind the scene as always, but he keeps driving every event, every responsibility which is given to him.
So I would like to express on behalf of
whole Institute of Science, I would like to express our gratitude to Professor Jagad Roy for organizing this event.
Thank you very much,
all of you. Thank you.
Thank you.